My name is Jordy Malaret, and I'm studying resistance to abiotic stress due to the overexpression of the SUPA gene in Arabidopsis thaliana. For some background on my project, the idea really came to me when looking at the threat that climate change poses to humanity as a whole, but especially within the area of agriculture and food production. As can be seen now, and as has been predicted for the future, as climate change worsens, we'll see this continued global temperature change, as well as more frequent and extreme weather events. These weather events include things like droughts, heat waves, and even change to soil nutrients, such as higher salt in the soil. These kinds of events stress the natural resistances that are inherent in crops and all plant life. And as has been seen, as these natural resistances are tested, it can result in things such as lower yields. And in the event that the natural resistances aren't enough for those stresses, it can result in the death of the plant. Taking into account those possible events, with lower yields and death, it can be predicted that humans may face some kind of food crisis in the future if we're unable to adapt our agriculture techniques and even the plants themselves to account for these stresses that climate change has created. For my hypothesis, I predict that the expression of the supergene is directly related to the resistance that a plant exhibits towards abiotic stress. Thusly, as abiotic stress worsens, the expression of SUPA will increase. Additionally, in Arabidopsis with mutated supergenes that overexpress the gene, resistance will be improved. For my methods to test this, I started with growing my Arabidopsis in two ways. Firstly, the growth of the wild type and mutant Arabidopsis, the mutant one having the overexpressed supergene, in soil for about a month, growing them to full maturity. I then also grew the Arabidopsis of both types in a sterile liquid media for 2 weeks to collect a larger amount of plant material for additional stress testing. For the stress testing portion, I would take a sample before the stress treatment. I would liquefy it, collect the RNA, store it, and then, in the case of heat testing, I would place the pot or the plant matter that I had pulled out from the sterile liquid media on a heating pad, bringing it up to 37 degrees Celsius, roughly 100 degrees Fahrenheit, to induce the heat stress response within the plant. I would then collect samples again after a 3-hour period and begin preparing for RT-qPCR. As I performed RT-qPCR to see more of how much of the gene is being expressed in terms of production of proteins and such, I used that to determine the expression levels before and after stress treatment, and then was able to then compare across the mutant and wild type to see that difference. As for the results I found, on the graph on the left, we can see how M-sulc is this overexpressed line of SUPA, and this difference between the two bars shows that in the event of the same kind of stress, the overexpressed SUPA gene was able to express at a significantly higher amount than was the wild type, which was that correlation that I was ultimately looking for in my RT-qPCR. And my data on the right is just the numerical calculations I took to get to those fold expression levels, which is the level that they're expressed compared to the control. For further research, I hope to accomplish, and would be something that would be great as an additional part of my project, would be determining the survival rate across wild type and mutant superlines in response to various abiotic stresses, being able to test them on more kinds of stresses, as well as being able to replicate more realistic weather events such as longer droughts, longer heat waves, things like that. Try to make it as close to a realistic situation, and then really observe what is going on within the plant on a genetic level, as well as an exterior level in a morphological sense. And then also being able to compare genetically modified super overexpression lines to the wild type super sample, as well as the mutant sulc samples. Um, this would help determine if the effectiveness of an artificial modification as an approach to addressing food security in crops, as we can then compare it to that natural mutant of SUPA. Thank you.